Sagan Lecture. Paul is, in many sense, an ideal model of a biogeosciences scientist in the modern world. He grew up in New York City. Maybe that's not the model, but he grew up in New York City, got his PhD from University of British Columbia, has held a variety of positions, but some of the longest term ones were in the scientific staff at Brookhaven National Lab, and more recently as the Board of Governors professor at Rutgers. He's a member of the National Academy of Sciences, a fellow in American Geophysical Union, the American Academy of Arts and Sciences, and the American Academy of Microbiology. When I say that Paul is really kind of the archetypal specimen of a modern biogeochemist, that reflects my admiration of his ability to move from the molecular to the global, from the lab to the field, sometimes on long oceanographic cruises, from the Precambrian to today, the kind of interdisciplinary and integrative work that makes the biogeosciences and global change ecology so exciting. Most of his work is focused on nutrient uptake and photosynthesis in marine phytoplankton, in which he has tried to sort out through time the role of nitrogen and phosphorus and iron and silicon. Much of this has been done at the physiological level, but Paul has been so bold as perhaps to produce the best estimate of global marine net primary production, published a few years ago in Limnology and Oceanography. Now, it's particularly interesting today. I went to a prep school where you could get kicked out for almost anything, missing class and smoking and drinking or whatever. But in the back room, we had this book called The Story of O, well thumbed, kept from the headmaster at all costs, and kind of sneaking through that. And what it is now, 45 years later, maybe nearly 50 years later, a long time later, that I find myself introducing a speaker with the title, The Story of O. And I'm sure this will be an entertaining talk, and he's going to trace the evolution of the Earth's oxygen cycle and its relevance to some of the earliest life on Earth and outside the solar system. So please join me in welcoming this year's Sagan Lecture, Paul Falkowski.
Thank you very much. Thank you very much. Thank you very, very much, Bill, for such a wonderful introduction. I just want to preface this. I don't have billions and billions of slides. I want to talk today in a sort of a serious way for a minute in the introduction. Next year marks the 150th anniversary of the publication of Origin of Species, and there will be many, many, many celebrations around the world of Darwin and his mark in science. Those of you that know a little bit about Darwin realize that his idol and one of the people that actually supported him for his election to the Royal Society was Charles Lyell, sometimes called the barrister geologist, because he was originally trained as a barrister, and he was one of the most lucid writers of science in the 19th century. And the reason I want to talk about Darwin for a minute is that Darwin was struck by a basic problem in science, and that is the origin of life, really. And he really drew the first tree of life. In fact, it's the only drawing in the Origin of Species is a tree of life. It's 150 years later, and we still don't have an answer to that question. So if you think about the two fundamental questions that face us as human beings, that I think we ask when we're 5 years old, it is, where did we come from, and are we alone? And I think we'll answer the second one before the first. I think that we have the technical capability at least to scan extraterrestrial planets, and extrasolar planets, and look for marks there. I'm going to deal with that at the end. But the origin of life remains one of the big enigmas of science, which most of us have avoided quite successfully in our careers. Now I'm going to start talking today about basic biogeochemistry. So most of you are familiar with this. We cut our teeth on these kinds of diagrams. This is right out of Bill's book, but he didn't make the figure. The figure is an old figure. And it's really the distribution of elements in our neck of this galaxy, right? And if you think about it, we have several elements here. So there's hydrogen, carbon, nitrogen, oxygen, phosphorus, and sulfur, which form what we will call the big six elements. Those are the elements that make up the macromolecules of life. And those are all formed in the main sequence in stars, obviously. And so their chemistries are very interesting. They're coupled in a very profound way. So these are the main elements that we think about in origins of life. And the one element there that's a bit of an anomaly is phosphorus. Phosphorus can undergo redox reactions, but the primary reaction of phosphorus is acid-base chemistry. All the other reactions of the other elements are redox reactions. Now, here's where geochemistry and biology have parted ways, if you will, very early on in the history of both sciences. Biology is the study of electron transfers at its core, right? That is the metabolism of life. Every organism on the planet, every single organism on the planet exchanges gases with the environment. So the planet is composed of two major geophysical fluids, the ocean and the atmosphere. And those geophysical fluids serve as a conduit to move electrons and reducing equivalents around in gas phase, for example, or in gases solubilized in water. So, for example, let's just take the very, very simple, obvious situation. We're all in this room. As we are all sitting in this room now, we're all breathing in oxygen. The oxygen isn't made locally. It's not made in this building. It's not made in the streets of San Francisco. And it's not made very much at this time of the year right outside in California. So the oxygen is actually coming from a remote part of the planet that was made maybe years and years and years ago, maybe thousands of years ago, and has been now available and accessible to you and me and to every other organism on the surface of the planet that is consuming oxygen. So that makes it a very, very fundamental issue of the generation of a gas that can be used as an electron acceptor universally by common metabolism. And that is one of the most fundamental processes that allows coupled cycles on geological timescales. So life is fundamentally electric. That's actually quite obvious, I think, but we don't think about it this way. Basically, you and I are human beings, and every other organism like us, like E. coli, because we're just basically fundamentally E. coli that have grown up with some eyeballs. We fundamentally are fuel cells, okay? So we derive our energy for growth by moving electrons from a substrate to a product, and the substrates and products have to be cycled. That's quite obvious. So with all due respect to my friend Joe Kirschvink, who's in the audience, if the Allen Hills meteorite had a single organism in it, one organism, and it was the only organism ever on Mars, it was doomed to extinction. You have to have microbes that are like lawyers. You can't just have one. Lawyers, if there's one lawyer in a city, he will become extinct. Okay? Now that may be a very good thing. But we produce a lot of lawyers, and we also produce a lot of microbes, and they obviously operate in cycles. And in fact, I think when the people that are going to make the most money out of the bailouts are going to be lawyers, right? Anyway, so biological processes are ultimately paired. And in laboratory settings, This is what you will find your friendly neighborhood biochemist, or now that's not de rigueur anymore, so we call them what, chemical biologists or biophysical molecular ecologists or God knows what. But anyway, this is what they study. They study oxidation reduction reactions. So in oxidation reduction reactions are the movement of electrons with or without protons, in case you forgot. There's the trivial equation, which is how we get oxygen on the planet, right? The trivial equation is the oxidation of water by the energy of the sun. So this is the only reaction in biology where you do not take, you make bond energy from a source of external energy, not from preformed bond energy. All right? It's the only reaction on the planet that does this. We don't have a biological system that transforms radiogenic energy into bond energy, or heat energy into bond energy, or wind energy into bond energy. It's just solar energy. So photon conversion to an electron driving force is the core of the reaction, and I'll explain that a little bit more deeply in a second. And this is coupled ultimately to the reduction or putting electrons and protons, not hydrogen, the equivalent of hydrogen, but we don't put hydrogen. So the reaction of photosynthesis is taking water and splitting it into these equivalents. There's no free hydrogen that is made in the photosynthetic reaction. It can be made de novo in an artificial system, but it cannot be made biologically this way. All right, so now we take these electron equivalents, we couple them to CO2, and we make basically high energy molecules, carbohydrates, lipids, and so on, which serve two functions, right? They serve a function as building blocks for you and I, and they serve function as an energy carrier for all other life on the planet. Now, they're obviously, in a test tube, when you do these kinds of things, those reactions are coupled. The trick of the story of O, of course, is that we're breathing oxygen, so there must have been a process that didn't immediately consume it. So every geochemist is trained very early on that oxygen, free oxygen on a planet, has to be associated with the burial or removal of reducing equivalents somewhere in the lithosphere. So we usually think about this in terms of removal of organic carbon and storing it somewhere in the lithosphere, and or sulfur equivalents, because you can also put electrons onto sulfur. So oxygenic photosynthesis and aerobic respiration, by definition, cannot be coupled on geological timescales if we want to generate oxygen. Basically, we have to take and put this juicy molecule somewhere out of sight so it doesn't go into a respiratory chain. So that's relatively, again, standard operating procedure for a geochemist, and we all probably have learned that. So the electron marketplace is really a very interesting area that I explore a lot, and I've tried to understand how the redox couples have occurred, and I'll explain that in a few moments. But in the earliest portion of Earth's history, probably for the first 2.3, 2.4 billion years or so, most of the solar energy that was converted into bond energy did not use water. It used H2S, or it used other sources of electrons, such as iron. But water is actually a very difficult thing to split, to oxidize. Now, today, virtually all of the carbon that is fixed on the planet is coming from the splitting of liquid water. And that innovation, biologically, is one of the most enigmatic in biochemistry and molecular biology. And it is the equivalent of, if you will, Darwin's eyeball problem. If you're familiar with the history of evolution of Darwin, Darwin was very, very, very concerned about how did the eyeball emerge. And we are only beginning to solve that problem. So the evolution of a nanomachine is what I want to point you to at the moment. Now, I'm just going to show you this cartoon. It's a cartoon. It's for heuristic purposes, I think it's instructive. There are two light-driven reactions in all organisms that oxidize water, two. And the first one is called Photosystem 2, and that's because it was labeled that way by a British person, right? So you named the first one Photosystem 2. Okay, so the second one became Photosystem 1, and they operate in series. So let's just talk about these photosystems for a second. These nanomachines, as I will show you, are extraordinarily conserved in biology. And this machine over here is an ATPase. This is what gives you ATP, what gives me ATP, what gives E. coli ATP, it what gives everything on this planet ATP. And it works in an amazing way where protons go through a little tube here, and as they go through, these little machines on the top, they actually whirl around. They actually turn physically. And they generate ATP molecules from the protons. Okay? This machine over here splits the water. It has at the bottom of it a series of manganese atoms, 4 of them, which I'll show you in detail in a second, that are coupled to 2 major proteins called D1 and D2. They were named this way by Namhai Chua at Rockefeller University for diffuse band 1 and diffuse band 2. These are the 2 oldest proteins known for splitting of water. There's no other mechanism. We know where these derived from. These were derived from purple, non-water splitting photosynthetic bacteria. This machine over here, it was derived from green bacteria, green sulfur bacteria. How the 2 machines came to be in one organism is totally enigmatic. Nobody knows. All right. Now, for 25 years, people have tried to determine the structure of those machines. And in the early part of the 20th century, Horst Witt in Berlin and Petra Fromme at Arizona State, who is now at Arizona State, who was in Berlin in his group earlier. Horst died last year. Worked very, very hard to get resolution of these proteins at 4 angstroms, which is not a very high resolution. Subsequently, there's been a lot of work to beat those down 1/10 of angstroms at a time, and we're down to around 2.8 angstroms resolution, so we know what these proteins look like in some detail. And here is a face-on view of the manganese cluster. So there's manganese 1, 2, 3, 4. And those are, we know their coordinates very well. Now here's an amazing fact as we step, okay? We know the structure of this protein at 2.8 angstroms. We know that it produces all the oxygen on the planet. You know something? We don't know how it works. We do not know exactly how water is split. If you think about it, it's probably, we know quite a lot of details about side pieces of it. We know that one water molecule at a time enters a little canal, and electrons are extracted, 1 photon, 1 electron, and the intermediate O. There is really, in the story of O, there is no such thing as O. O doesn't exist as a free atom. O can only be O2. So the first O is actually coordinated to a manganese-calcium complex until the second water molecule comes through, and then the 2 O's are put together, and O2 is released as a byproduct, as a gaseous byproduct, diffusing freely out of the cell. And that is how that works, but we really don't understand how the electron is moved from the waters to the manganese to what ultimately becomes electron acceptors. Now, nature invented this, and it occurs in a single clade, 1 single clade of bacteria, in the cyanobacteria. That was the sole time that invention occurred that we know of. It doesn't occur in any other prokaryote. This oxygenic photosynthesis reaction is a single one-shot deal. It was made once. Nature did something very interesting to spread the risk around, and it took that machine from a cyanobacteria and it allowed it to be incorporated into other organisms wholesale. So there are 2 machines that were incorporated wholesale into other cells. The first is the mitochondria. That's what powers our energy supply, you and I, and every other animal on the planet, and every plant on the planet. The second was the chloroplast, the plastid, the photosynthetic machine. And that was incorporated several times, and you can see the initial event was an incorporation way at the top to an organism that already had a mitochondria. When this occurred, we're not sure. But it most likely occurred about 1.5 or so billion years ago, in the mid to late Proterozoic. The cyanobacterial origins are also very poorly constrained, I should point out at the moment. There are arguments back and forth as to whether it's 2.8 billion years or 3.2 billion years or even earlier or even later at 2.5 or 2.4 billion years. I don't want to go into that issue, but it is a very, very interesting problem that we haven't constrained that better than we have. Anyway, this becomes now a new type of cell, which is a eukaryote, which is the origin of plants. And all plants are derived from that single transfer, that single appropriation. This is what is called the endosymbiotic theory, or the endosymbiotic association. And just as an aside, historically, it was popularized mostly by Lynn Margulis, who wasn't the discoverer of it, but she popularized this notion. She didn't name it. It was named by Max Taylor at the University of British Columbia, but she did have this notion down pretty early on. And she was married, of course, to Carl Sagan for many years. So that process has been repeated many times in many different symbiotic associations have emerged, and that has led rise ultimately to all the different clades of eukaryotic algae that are in the modern ocean, such as diatoms, which are secondary symbionts, or coccolithophores, which form the chalky deposits of the cliffs of Dover, for example, in geological time, or to dinoflagellates, which often cause red tides. And so these are all multiple association events with cells that were originally not photosynthetic, that became photosynthetic through the engulfment and retention of the photosynthetic apparatus. And that is how nature has spread the risk. So we have never lost this machine. This machine is identical in every one of these organisms at its core. All right. Now, across the tree of life then, as I said, the cyanobacteria, the guys in yellow will be the oxygenic guys. They're the only guys. They're related to the plastid down here, the chloroplast, and they are clearly the origin of plastids. And they have been spread throughout the entire tree of life, but only through the eukaryotes. Only through the eukaryotes, which tells you that the number of genes that are required for this is much too great to allow simple lateral gene transfer across the clades within the bacteria or across from bacteria to archaea. So whereas nitrogen fixation is only found in a prokaryotic system, that is spread all over the tree of life down here because it was laterally transferred, not so photosynthetic machine. Now, I just want to discuss this last bit of piece of molecular information with you. It's what I call the frozen metabolic accident problem. And that is, there is a notion in biology, which is wrong, that things are always going to operate at their maximum efficiency, and they will be selected through time to operate at the maximum. And that's just simply not true. And I'm going to point this out to you. Here is a classic example of just getting the job done, even though the machine doesn't work very well. This is a photograph of an autoradiogram. So this is radioactive sulfur-35 labeled proteins. They were chased with cold sulfur in the second lane here. So we labeled these up with hot radioactive sulfur, and then placed the cells in and isolated the proteins, ran them out on a gel, and did the same thing after we chased it with cold sulfur for several hours. And what you can see here is, you see how this band just virtually goes away? That protein is totally turned over. It's there, the protein is there, but it's no longer radioactive. It has gone through several cycles, actually. That's that D1 protein. That's that diffuse band 1 protein that's one of the core proteins of the reaction center of photosystem 2. That protein in every photosynthetic organism, bar none, has a turnover of once every 30 minutes. That means that every 30 minutes, every one of those proteins is replaced with a new one that is made in the cell just because that protein has been damaged. Now that's amazing. Here you have a machine that is so old, and yet nature has never been able to figure out a better way. What it did was what I call the Microsoft problem. It just took a whole bunch of code and went and made a repair cycle where it just made just don't worry about it. The old thing works. Just make another one and stick it in there. Fix it, This is exactly what Microsoft does, and it worked for Microsoft and it worked for biology. And this isn't the only place like this. There's many frozen metabolic accidents in nature. And just to point out to you this, these are unbelievably conserved. This is the rate of substitution of this protein. This is PSBA. This protein is conserved at 86% at the amino acid level from a cyanobacterium to an oak tree. Okay? So nature has done every site-directed mutation on that you can imagine and then some, and yet that protein still only has a life expectancy of 30 minutes in the cell. And the reason is a very simple one. When you have proteins that have to interact with each other and have to interact with lipids and have to interact with prosthetic groups like metals, there isn't a lot of room to start monkeying around with mutations. You make a little mutation here, a little mutation there, and pretty soon the thing doesn't work anymore. And that's the conservation problem. So it comes from, and this is a map showing the integrated outcomes of essentially the predicted interactions of proteins with each other in all these complexes from the work of my former student, Tuo Shi, for his PhD. But you can see that these interactions between D1 and D2 and these other proteins and so on are extraordinarily strong, and that's why the conservation of these machines is so high. Now, these are the outcomes of these guys. So these are dinoflagellates. That was a diatom, for example. And what is the outcome of all of their carbon fixation ultimately? So they can split the water. In the modern world, productivity of the ocean, about 25 to 30% of the productivity of the global ocean is dominated today by diatoms. They sink. And they sink into shallow seas. And in the shallow coastal environments around the world, not everywhere, but in many environments around the world, they form part of the organic matter of sediments. And that organic matter of sediments in coastal shallow seas becomes the repository, ultimately, of carbon through time on geological timescales. It's not the deep ocean. But more importantly, it becomes the generator on geological time of oxygen for the planet, right? So that's a main generator. Now, there's a second problem in oxygen, which is actually a very interesting one. So if we had an Earth without continents, simply the tectonic subduction of that organic matter would put it back into the furnaces quite quickly, on order of a few 100 million years, and the net oxidation of the planet would be relatively minimal, slow. If you want to take that organic matter and you want to really store it somewhere, you've got to get it out of the Wilson cycle. Now, when Twelzo Wilson conceived the cycle, I don't think he was thinking about the oxidation state of the planet, but it really is a very interesting problem, and it is because of tectonics that we are allowed to have such high oxygen concentrations. Effectively, what happens is in the latter stage of the Wilson cycle, which we will enter in the Atlantic Ocean, for example, maybe within the next 50 to 100 million years, the way I pointed out is, unfortunately, California is not going to meet Japan. New York is going back to France. Okay, so the French. Get a job in the CNRS. I'll learn my French. Okay. Anyway, as we're in this stage of the Wilson cycle for the Atlantic Ocean right now, right? So as we start to cool and subduct along either one or both of the present passive margins, we are going to get some accretion, or actually it's just an addition of essentially shaving off bits of sedimentary crust. It's really part of the craton anyway, but it's going to be part and uplifted onto the craton out of the Wilson cycle. So we are going to be taking this carbon and putting it into it's my own phone. Sorry. Putting it onto continental cratons as an orogenic event, right? So the west coast of the United States, for example, has seen this, and once you do that, you can store this organic matter in a much more permanent way than you would if you just had it in the seafloor, in oceanic crust. So really it is the rise of continents and the Wilson cycle which allows a long-term accumulation of oxygen. So you have two events, right? You have the evolution of a process that could split water, coupled now with the burial of organic matter and tectonic processes that allow the uplift. A rather amazing, Lee Kump doesn't like the use of the word contingency, but I suspect that it is rather amazing that that happens on this planet. It's not going to be a very, very common feature of terrestrial planets, I don't think. Now, I just want to go into a very, very brief period here where I'm going to talk about the ball in the bowl metaphor for the first half of Earth's history. So the first half of Earth's history was fine without oxygen, frankly. You know, most microbes just did their thing. They had their cycles. They just didn't worry about oxygen. And the reason I'm talking about this ball in the bowl is that the ball was hit several times. If you just think about it as the system of life. We had bolides, we had goodness knows all what. Very difficult to reconstruct that period of Earth's history because there's so little rock volume. But there were many traumas, I'm sure. And the ball stayed in that bowl. The second half of Earth's history, and by the way, I call the first half of Earth's history the research and development phase. Okay? That's when biological innovation really occurred. The second half of Earth's history was basically just copying what everybody else did and just putting it in different forms. So the engines of life were invented in the first half. In the second half, all you did was take the engine and put it in different models of cars. All right? So I'm fundamentally correct when I say that you and I are really not much different from E. coli. Okay? And E. coli will outlive us. So we're the fragile species here. All right, now the second half of Earth's history, we push this ball into the second bowl, which is a bowl with oxygen in it. And here is the amazing thing. Once that happened, it didn't go back. This Earth became oxidized. So now let's just think for a second. I mean, this to me, I don't know, is rather mind-boggling. There are two major gases on this planet, broadly writ. There's N2, which was probably here since the beginning of time, virtually always is N2, and O2. O2 is created totally biologically. So 21% of the volume of this planet in gas, 21% is a biological outcome of the planet, which is, if you think about it, amazing. That it has remained and been sustained at such high levels for so long. Now, we will never go back as a planet to a point at which we will be reducing again. At least not by any imagination that I have. I can't imagine how that could happen. Photosynthesis is here to stay as long as water is on the planet. So as long as we have liquid water, we will have oxygen on the planet, and we will never go back to that first initial first 2.3 or so billion years of Earth's history. I don't know if you can see this, but you can certainly read about it in Science. Tom Fenchel and Ed DeLong and I we desired to do one very simple thing. We just wanted to write a sort of a wiring diagram of the electron transfers that take place on the planet, and this really is a metabolic map of life, stripped to the core. Stripped to the core. Now, I could have put gene numbers on there, and we could have just had annotations as if it were really a metabolic map. The point I want to make to you is, in this diagram, there are approximately 1,500 genes or so that are essential to life on Earth. The rest of the genes that we report in Science magazine or in PLOS as the 7 million new genes that have been discovered in the ocean, for example, by my colleagues at the Venter Institute and others, those genes are looking at paint chips. They're looking at the color of the car. They're looking at the size of the mirror. They're looking at the design of the fender. These genes, the genes that run these cycles here on this graph, are the ones that are really important and the ones we should be studying. Those are the ones that we should be looking at most carefully, because their understanding of them will change our world for our children, I assure you. If we can split water to make hydrogen, the world's energy game is different. If we can fix nitrogen, the world will be a different place. So anyway, it looks a lot like this. And in some respects, this is a more complicated map. But the idea is the same, that organisms are like electrons, and they travel like people in a circuit. And occasionally they go from moving an electron from one place to another, from the red line, for example, to a green line to a yellow line. And that is the nature of the intimacy of the coupled electron transfers that make this planet so incredibly robust geochemically. So let's continue on. I want to talk a little bit about the oxygen problem and the nitrogen crisis, which I thought about several years ago. We've written about it and we're working on it, and I think it's a very, very interesting problem. So of those big six, there's one other element out there that's really, really important. Really important. Without it, we don't have any proteins or nucleic acids. We're just a bunch of fat, okay? So if you want to go on an Atkins diet, you need this molecule, okay? You need ammonia. Now, all of the nitrogen on the planet, that for any intents and purpose, virtually all of it is in the atmosphere. I'm not going to talk about the deep lithosphere and so on. There's a lot of nitrogen potentially in the deep lithosphere too. But for the most part, the inorganic phase of nitrogen is N2. And N2 is like a noble gas. N2 is very unreactive. In order for the nitrogen cycle to work, we had to make three inventions ultimately. It's all a very interesting one. So N2 was reduced to ammonium. We're adding electrons to it to make this. That's good. That happened very early in Earth's history. Very early. Then, once that happened, the cycle could be okay. The first half of Earth's history without oxygen, you just had ammonium. That's the end of it. It just goes through tectonics, comes back out ultimately as N2, or even as ammonium, it doesn't matter. But that would be the cycle. Very slow, but it works. In the presence of oxygen, and only in the presence of oxygen, can you create this reaction. You can take ammonium and drive it to ultimately a thermodynamically stable form of nitrogen, which is nitrate, which is what most of the deep ocean, the form of nitrogen in the deep ocean today, right? So this is the process that is again catalyzed by organisms, by bacteria. And this is an oxidation reaction. So this is a reduction reaction, an oxidation reaction, all biologically driven. And then we take the nitrate, and in the absence of oxygen or very low concentrations of oxygen, we denitrify this. And this is a bunch of organisms. They dump electrons because they don't have oxygen. They look for some place to dump their electrons, and they dump it onto nitrate, and they produce a little bit of nitrous oxide or sometimes nitric oxide, and ultimately going on to N2. So this is laughing gas right there. That's laughing gas, right? And this is what Viagra does for you, right? So the NO is what Viagra does for you. So they're very old molecules, important ones. So that's a reduction reaction again. So you have a reduction, oxidation, reduction, all biologically driven. Okay, fine. Okay. I don't want to bother you with flute music, but we can couple these in a very nice way. And all I want to point out in this particular part of the talk here is that the reduction of N2 is essentially a respiratory-like reaction. We're taking a carbon molecule with hydrogen on it. This is a sugar. And putting these electrons and protons onto the N2. So in effect, I want you to think about nitrogen and the early Earth as an electron sink. That's what a major role of that molecule was. It was a major source, a major repository for electrons. Excuse me. This is nitrogenase. And the reason I'm putting this up here, it's another interesting molecule. This is a very old molecule. It does the first part of the reaction of taking the N2 and making it to ammonia. Very important reaction. Okay? We have to do this to make our own food, but we don't use this molecule. We use the Haber-Bosch reaction. And the Haber-Bosch reaction is a high temperature, high pressure reaction with a catalyst. We take H2 gas under high temperature and pressure, and we put it in with N2 gas with a specific set of catalysts, and we make ammonium. That's how we make food. Plants have done this with this enzyme. Now, we know this enzyme's structure at 1.3 to 1.4 angstroms resolution. It doesn't get any better than that in protein chemistry. Another dirty little secret. We don't know how it works. So here are the two main reactions that run the world: oxygenic photosynthesis and nitrogen fixation, and we don't understand the molecular mechanism by which they work. And I find that pretty fascinating. So I would hope that, somewhere before the next 150th anniversary of Darwin, we've answered that problem. I'm sure we will. Phone rings, microphones fall. Okay. All I have to do is fall up the stage. All right. Let me just take you through what I call the nitrogen crisis story, which is an interesting one. So this is a Black Sea profile. Black Sea is a very nice place to go. You can get rugs there quite easily, and nice tea, good music, nice barbecue. But you can also actually go out on the ocean and do a few profiles in between the rugs. So anyway, bugs are produced up here in the upper euphotic zone, which has got oxygen in it, and that's the oxygen profile here. Okay, and then these bugs sink, and the deep portion of the Black Sea has no oxygen in it, right? Because the salinity of the Mediterranean inflow waters is extraordinarily high, and it's capped with these fresh waters coming down the Danube and other rivers into the top. So it's like a Napoleon. The whole Black Sea is set up like a Napoleon, extraordinarily stratified. And what happens is this ammonium then diffuses up, and it hits this oxidized world where the nitrifying bacteria are, and so they make nitrate. And then some of the nitrate diffuses down to this suboxic world, and it is denitrified. And this is the excess nitrogen over and above that of the atmospheric equilibrium. That's denitrification, okay? But look over here. So there's this place where the ammonium is going up and the nitrate is coming down, and there's a node. There's no fixed inorganic nitrogen, virtually none at that spot. And if we take that profile and turn it on its side, this to me becomes an image of the Archean to Proterozoic transition. If you're going from the first ball in the bowl, that ball has to move from this place to that place. Now here's the problem. If you denitrify the ocean, You just take away one of the most essential elements for carbon burial. So that's a very strong negative feedback. That pushes the ball back into the first bowl. That prevents the atmosphere from getting oxygen. Interesting. So here's a feedback that I hadn't thought about until, maybe after a couple of beers in Istanbul. But we made a very simple model. By we, I don't do this for a living. So I went and described this in words to Mick Follows. And Mick made a model, and it worked, but then Katja said, let me try it, and she did it and fixed a few things, and it worked really well. So we use this kind of very simple model. I like simple. And in this model, we put the flute music I showed you in a differential form, just as a derivative of time. And what the model does is this. It just takes N2 from the atmosphere. It allows it to be fixed to ammonium. It produces organic matter, which sinks and is buried. And as it's buried, then we can produce oxygen. If we produce oxygen, we can take the ammonium and go through the whole cycle. We make nitrate. If we take nitrate, we can, depending on the oxygen concentration, denitrify everything back to N2. And the oxygen can be produced or not, depending upon what nutrients are limiting in this ocean, right? There's a tipping point somewhere here of around 15 micromolar oxygen. At around 15 micromolar oxygen or above, nitrate is stable. And you then can enter an oxidized world where oxygen can be produced continuously forever. If below that point, if you're here, denitrification wins. And you're pushed back, the ball goes back into the first bowl, and you don't make oxygen. You stop. Okay, so let's take a look at it. This is millions of model years. And essentially what happens is you see very, very quickly, within a few million years, you denitrify the entire ocean. We're down here, the N to P ratio, this is N to P ratio is 0, effectively. There's no nitrogen, no fixed inorganic nitrogen in this ocean. You're fixing nitrogen, but as fast as you fix it, the little bit of oxygen that's around is converting it to nitrate. The little bit of nitrate that's around is denitrified back to N2 gas, and the amount of fixed inorganic nitrogen in that ocean is 0. And we do some tricks because it gets very boring. You can run this model forever and never escape from that. And what we do is we add a bunch of phosphate, we change shelves and so on. But essentially, the end of the game is this. If you want to escape from this, you have to produce a lot of very shallow seas. Because very shallow seas become the repository of organic matter. So here's a very interesting problem. What was the sea level like, and what was the topography like, the submarine topography like, during the late Archean, early Proterozoic? We don't know. But that's an interesting problem. The phosphate story determines how much oxygen you get, not the rate at which you get it. The size of the shallow seas determines the rate at which you get it, not how much you get. All right, so anyway, the story is that the oxygen cycle is constrained by the nitrogen cycle, and the nitrogen cycle is driven by the oxygen cycle. They're absolutely dependent on each other. It's a very interesting problem here of at least 5. The 5 big molecules that I told you, except for phosphorus, are all interrelated with each other. And figuring all those redox reactions out is quite a story. Now, this is a kind of, I just want to skip this to, okay, that's a model. Models don't prove anything. So we've been working for several years, Tracy Kwan in my group and Linda Godfrey with the good graces of the Agaron Foundation, who have funded us, and NASA, who have funded us over the years. We've been working on very simple analysis of isotopes. There are only 2 isotopes of nitrogen, stable isotopes. Nitrogen 15, about 0.3% of the total mass, and 14, of course. And the idea is this, that if you denitrify, you're taking the light isotope of N14 and putting it back into the atmosphere. What's left behind is heavier isotope in the organic matter. So as you add a little bit of oxygen to the system, a little bit of oxygen, things should get heavier and heavier as you denitrify. And once you get above some concentration of oxygen, you get that nitrate stability, and you will then go back down into a world in which you can go back down into a world in which you can have stable isotopic values that can be identical on either side, by the way, so not linear, obviously. So you have to know approximately how much oxygen there is from other proxies. We use metals, but there are many other ways of doing this. So I just want to march you very quickly through this. These are Toarcian shales. They've been worked on a lot by us, well, I'm just going to show you the data of Hugh Jenkins at Oxford, who worked on these shales quite nicely several years ago. Hugh had shown these Toarcian black shales really were a very, very widely distributed. So there was, during this period, this is Lower Jurassic, 193 million years ago, this period of Earth's history, the ocean was anoxic. For reasons we don't know, but just it was, okay, or very low oxygen. And watch what happens. So I'm just showing you that we can go to a place in Earth's history where we can make these kinds of measurements. And here is the nitrogen isotope data, and it gets heavier. We denitrified the ocean for several tens of thousands of years, probably, if not more. Okay, that looks good. You can use the isotopes to at least say whether or not this model that I showed you earlier is really true or not. Tracy then goes, we get rocks from Mingelsheim, which is a portion of the Tethys Sea, during the TJ boundary. So this is 200 million years ago, almost exactly on the button. There's the del N carbon-13. And there is the little spike. You see that right there? That's a 1.5 or 1 per mil change. That's the denitrification at that event. That's the TJ boundary, by the way. We know that from paleontology. It's not just, so anyway, from correlations. As we move up into the Lower Jurassic denitrified this portion of the ocean. Okay, that's interesting. Now let's go back deeper in time. These data are from James Farquhar. I like this data set very much. It's a little difficult to interpret sometimes if you don't remember how to keep track of 3 isotopes. But the point is this. This is probably, I think, most geochemists would agree today, that this is the best evidence for when we got oxygen on the planet, in the atmosphere. And it's based upon the observation that isotopic fractionation of sulfur was independent of mass of the isotope up until about 2.2 billion years ago, right there. This is the cap delta 33S is these green circles, and you can see this isotopic fractionation is quite ranging, far-ranging, from early Earth history all the way up until this, and then all of a sudden it stops, and it never reappears. And the interpretation that James and Mark Thiemens had is quite elegant. Sulfur has a UV cross-section at about 185 to 215 nanometers. So the atmospheric reactions of sulfur with UV light were quenched at about this time. The most likely quenching agent had to have been ozone. So in order to produce ozone, you need molecular oxygen. So the argument goes that this is a point in time where there was sufficient molecular oxygen to produce ozone, which led to, forevermore, a mass-dependent isotopic fractionation. So the mass-independent isotopic fractionation ceases. The mass-dependent isotopic fractionation is what happens for the rest of Earth's history. All right, now let's go back here and continue. I'm sorry. So these are our isotopic records of nitrogen. I'm not going to go through these. These are data that we have all over the map. Some of these data are really, really of poor quality. I'm not going to name names, but the data right here are very, very clearly altered diagenetically. These are way too heavy for biological processes. We've gone to a craton in South Africa, which was drilled by the Agaron Group, and through the good graces of Joe Kirschvink and others. We've had access to cores that have been analyzed by several other people within the group, and we've analyzed the nitrogen isotopes. So this portion of the core is it's before that so-called great oxidation event where the mass-independent isotopic fractionation disappears for sulfur. It starts at about 2.75 or so billion years before present, and this is going up to roughly about 2.45. And what you can see, this is the nitrogen isotope now composition of kerogen. So this is purely organic nitrogen isotope values. And you can see that there are places here where these isotopic values go up for several tens of millions of years to values of approaching 8 or so. I would submit at first blush that this is strong evidence that the hypothesis that I presented to you earlier of this nitrogen crisis preventing the oxidation of the atmosphere is real. So the coupling of the nitrogen cycle to the oxygen cycle is at present well supported by this and another core which we have subsequently done, which I'm not presenting that data for. All right, now let's get on to the end of the roadshow. And this, of course, is Bob Berner's plot with our data at the front end and Berner's data from many sources at the back end of the Phanerozoic oxygen cycle. It's a poorly constrained cycle. Oxygen is thought to be constrained at the high end by fire. I'm not sure that that's true. I'm not sure there's any constraint on the low end. So certainly fires can be a way of analyzing whether or not there was enough oxygen to support it, but I'm not sure that Belcher and McElwain's paper, for example, is really true that it's 15% is the lowest end during the Mesozoic, for example. Well, we can discuss that later if you wish. The point is this. Oxygen concentration somewhere in the Carboniferous rose very high. We think somewhere to the neighborhood of about 30%. They went down in the lower, we think they went down in the Mesozoic to somewhere in the neighborhood of around 13, 14, 12%, and have risen to the present values to around 21% over a long period of geological time. And I'm going to point out to you why that is. So let's just take a look. I like that picture. Isn't it cool? I mean, the Ichthyosaurus, largest eyeballs of any animal on the planet ever. Amazing. Amazing. It's not my phone. All right. So, several years ago, Mimi Katz and Alan Milligan and I and so on were working together, and we compiled first the carbonate isotopic record from cores. It was actually the first coeval isotopic record of carbonates for the Mesozoic. This only goes back to 205 million years. That's the end of the cores, sedimentary cores on Earth. I mean, that's basically where you stop. But this is the isotopic. We did this also for organic carbon in a subsequent paper. And I don't know, maybe you think I'm taking a liberty, but I think there's a secular trend. The secular trend is about 1.8 per mil over that window. There's a little bit of a wing at the end here, which is a very interesting one. This is probably almost certainly due to the rise of C4 plants on land, which changed the isotopic fractionation significantly. Now, what you can see over here is you can watch on this side, you can watch the opening of the Atlantic Ocean. So here's exactly, in my mind, this is what's happening. As we're opening the Atlantic Ocean, we're creating more and more and more passive margin, which is accumulating organic matter in this phase of the Wilson cycle. And subsequently, oxygen is being liberated to some extent. So the change in this isotopic value of carbon tells you we must be bearing organic matter. And if we're bearing organic matter, those reducing equivalents have to be reflected somewhere else as oxidizing equivalents. And the logical place is the atmosphere. So we constructed, for better or worse, an oxygen profile in percent oxygen for the last 200 million years. And I worked in a previous life in comparative animal physiology. It was my very, very first thing that I started to do in science under the leadership of Peter Hachachka at the University of British Columbia. But what I did was I started to think about the respiratory rates of animals. And here is the kick. So you go at approximately 55 million years ago from the PETM, the Paleocene-Eocene thermal maximum, from a value of around 16 to 17% oxygen all the way up to 23% oxygen. Now, if you talk to most paleontologists, vertebrate paleontologists, the end of the Paleocene really marks the beginning of the dynamic evolution of mammals. Mammals really start to get big and they start to diversify very rapidly. So it is the evolution of almost all of the major clades of placental mammals, large placental mammals. And here is where the rise of diatoms during this period, the burial of organic matter, and the opening of continental shelves and a passive margin has created an opportunity for what are, without doubt, we are the Hummers of biology, okay? It takes a heck of a lot more in calories to feed us, to generate our heat, and to produce enough oxygen for our offspring when they're in utero than it does for an alligator. Okay, an alligator can eat a chicken a month. Okay, it's fine. I mean, it'll eat you too if you happen to walk by, but if you gave it a chicken a month, it would be fine. Okay, it uses about 1/6 of the calories per unit biomass as a human being does. So animals like mammals require huge amounts of oxygen, and ultimately in every animal, believe it or not, in every animal, the oxygen that is used and consumed is restricted by diffusion. So you and I have far less capillary density at the end of our muscle tissues than a mouse does, and it scales to the 3/4 size approximately of the mammal size. So the larger the mammal is, the less capillary density it has, the less oxygen it can consume, but also the less oxygen is available for its placenta offspring. And that really, really, really constrains then the ability of mammals to operate at low oxygen concentrations and be large and placental. You can bear eggs at lower concentrations of oxygen. That's fine. Just keep them outside you. Or you can be small. But you can't get large, and you can't have a placental reproductive strategy without high concentrations of oxygen. So let me go back for 1 second. We live on a planet that is a very, very rare planet. It's obviously the only planet in this portion of the solar system that is in the habitable zone, really. Venus is too hot. Mars is too cold, although Mars may have been in the habitable zone some time ago. And it's a wonderful set of papers that Jim Casting wrote many years ago when he was working at NASA on this story as to why Earth is the way it is. So obviously, one of the drivers of NASA at the moment, and has been for a long time, is there life outside of our solar system. I'm not worrying about life on Mars, although we can get there and it's fun to drive rovers around. Essentially, the question is there life outside of our solar system? So we look for signatures, right? And this is where I'm going to end, because I'm going to come to the end, because this was the forte and dream, really, of Carl Sagan. This is really where Carl had such an incredible impact on science, our society, our kids. I mean, just, it was amazing. And Carl gave incredible lectures on this, those of you that remember him. We have several signatures of the IR reflectance spectrum, and the Galileo mission was one of the great missions that showed these reflectance spectrums early on, these spectra early on. So ozone is one, for example. Carbon dioxide and the CO2 spectra and the water spectra are obvious ones, and we can now just find water. Actually, just found water last week on a planet that I think was 6 parsecs away. So these are spectra. Okay, now the way we measure these spectra is quite interesting, and it's challenging. In the infrared, we actually are trying to null out the sun using an interferometer. You're nulling out the background star to a factor of about 1 million using an interferometer. That means flying satellites in formation. Very, very hard to do, with less than 1 centimeter wiggle between the satellites in formation at the L2 point. You're then staring at a planet and collecting photons at about 1 photon every 6 seconds or so, depending on the distance. Working at 3 parsecs versus 10 parsecs is a big, big, big difference in terms of the photon input. You're then scanning. So it takes you about a month to acquire a spectrum. That's the idea. It will take about a month to acquire a spectrum. But that's doable. And we would be looking for spectra that are indicative of life. So this is a very nice slide that Tim Lenton produced several years ago, and I like it quite well. And I think, as I was on the terrestrial planet finder team, what my role was there was to think of dynamic disequilibria in gas phase space. So oxygen's a no-brainer. If you had 10% oxygen, 5% oxygen on a planet, I say it's over. There's no way I can think of doing that without putting some life on the planet, okay? Maybe I'm totally out of it, but that's, to me, a really nice, simple sign. But before there was oxygen on this planet, for the first 2.3 billion years, what would you have seen? Well, then I would be looking for methane and nitrous oxide, right? Because if we denitrified the planet for several hundred million years prior to oxidizing it totally and producing real molecular oxygen, we would have been leaking that into that laughing gas out, right? So the laughing gas would have been a constituent that you could see in the IR spectrum. It's hard to see, but you can see it. Methane would have been one that we would be looking for. So those are the kinds of gases. You're looking at gases that are far away from thermodynamic equilibria. Okay? If you see ammonia and dimethyl sulfide, well, big deal. They're both reduced. But if you see an oxidized species and a reduced species coexisting, I can't think of any other way to do this except through life. And that's really where biosignatures are going to be an incredibly useful technique, coupled with incredible technologies from NASA, to develop these kinds of tools for answering the second question that you asked when you were 5 years old, are we alone? And I think we'll know that probably before my grandchildren are my age. I'm sure we will. So we're looking at the glows from other planets, and this is an artist's conception of the TPF. There are many conceptions. There's a Darwin mission out there from the European Space Agency. If TPF will get going again, or some resurrection of it in the new administration, when we can be interested in such things again, then I would look forward greatly to working with NASA and other agencies around the world to answer that second question. And with all of that, I want to thank all of my colleagues, and especially Linda and Tracy and Mick Follows and so on, who have been so kind to me and working with me over the years, and to the Agaron Foundation and to NASA and to the National Science Foundation to supporting my work. And I want to thank you all for being so patient and listening. Sure.
I think we have time for a question or two, and Paul's indicated a willingness to take them.
Norman.
In the front.
Carl Sagan is very concerned about the environment. Could you give some, you mentioned genetic engineering or molecular engineering of improved photosynthesis. Do you foresee a danger if we do that and get loose?
So Norman Sleep from Stanford asked me, do I see a danger of if these genetically engineered things get loose? Sure, but let's be realistic right now.
I know we can't do it.
Well, let's be realistic. I mean, many of you people, some people in this room like tofu. I don't particularly like tofu, but a lot of people eat tofu. There isn't a soybean made in the United States that hasn't been genetically modified, to start. Let's just get real, okay? So genetically modified food is there. What I'm talking about is taking a structure in molecular biological terms, protein structure, for example, and using it as a bio-inspired catalyst to try to make a catalyst. Essentially, all these are catalysts. They split water. If you go to your neighborhood chemist, and Steve Boxer would be an example in your institute, or John Brauman, and you said, okay, you guys are really smart. How come we can't split water? They don't know how to make the catalysts that do that. Now, Dan Nocera is trying at MIT. There are several people that are doing that. But at some point in the not too distant future, I would submit that somebody is going to take a bio-inspired molecule like the reaction centers of Photosystem 2 and make a manganese cluster that doesn't have the protein necessarily, that you just shine sunlight on it, and you will get a stream of hydrogen going one way and a stream of oxygen going another way. You can actually direct the molecules in a different way. And you will split water without the plant. Now, if that gets away, that's great. Let it get away. Let everybody have one of these. I mean, I think this is an amazing way that we could generate a huge energy carrier from photons. And ultimately, photons are the energy of the future. That has to be it.
And it's not in an organism, so we don't have to worry.
You don't have to worry about it. No, I'm not trying to create the eggplant that ate Chicago. Okay.
Thank you.
Another hand is very difficult to see hands from up here. How about over there?
You mentioned methane as an anoxic biomarker, but Titan has plenty of methane.
Yeah, it does.
So how would you know when you were looking at Titan and when you were looking at something?
You want to find a reduced and oxidizing species in the same planetary atmosphere. That's what I'm arguing, is that you can have reduced species. It doesn't tell you anything. Planets always will start out in a reducing environment. Always. They have to be, right? You can't start out a planet that I know of in an oxidizing condition. They can only become more oxidized through time. But if you produce species like NO or N2O in the presence of methane, those two species cannot exist in dynamic equilibrium without some kind of process like life that is extracting electrons from one species and putting it on, or removing electrons from another species. So there must be on those planets then an electron circuit that I would take as indicative of life. So yeah, Titan is not a candidate. Not a candidate.
One more?
Carl.
We'll pick on the front row.
Carl Pilcher is the head of the astrobiology program at NASA.
Paul, you didn't use the word habitability, but an awful lot of your talk was about what makes a habitable planet. And for example, plate tectonics was clearly involved
Sure.
in the processes that you described for supporting life on this planet. So what do you think are the minimum requirements for a habitable planet around another star?
Yeah, so the minimum requirements. My druthers would be, and I don't think that we could have habitable planets without there being liquid water. For at least at the surface may not be liquid anymore. So it is possible, for example, I could imagine, because there is subduction of ice on Europa. If you didn't have subduction of ice on Europa, it's a closed deal, right? But, and actually, I told this to Chris Chiba many, many years ago. That's when you can calculate almost exactly what the productivity of Europa has to be, because you know the oxidizing equivalents that are going down to first order. And if you know that, you know the amount of reducing equivalents that can be exchanged, and you can calculate the productivity of Europa. So those kinds of planet systems will be hard to detect because there's no atmosphere. But if you have an atmosphere with water, and you have an atmosphere with at least gases that are composed of the light 3 or 4 elements. So I'm looking for gases that will be with hydrogen and carbon, hydrogen and nitrogen, nitrogen and oxygen, and carbon and oxygen. Those gases out there, I think, would be extraordinarily useful signatures. The sulfur gases, you can get into a strange space, because sulfur disproportionation doesn't have to be biologically driven. You can have sulfate and sulfide that exist very happily in a world without life. I'm pretty sure that can be sustained for a very, very long time, and the atmospheric residence time of these would be relatively short anyway.
Paul, thank you
Thank you
for
very
a thoughtful talk, and one that will cause us to think.
Thank you very much for inviting me.